Hi, everyone. My name is Mathew. I'm a graduate student at the University of Virginia, and the title of this talk is Transcriptomic Response of Brain Tissue to FUS-mediated BBB Disruption Depends Strongly on Anesthesia. So significant effort has been placed in trying to understand how FUS experimental parameters contribute to cellular responses in the brain. Some examples include peak negative pressure or microbubble dose. Here, we argue that anesthesia is also a parameter that should be considered. If you take a look on the table on the right here, you can see a meta-analysis from our lab where we analyzed about 60 FUS studies across multiple disease models and therapeutics and identified a wide variety of anesthetics that were used. We categorize these into isoflurane-based studies, ketamine with an alpha-2 agonist, or miscellaneous, which are various combinations of other anesthetics. And this is important to highlight because these anesthetics vary widely between each other in their effects on the vasculature, neuronal signaling, and inflammation, as well as metabolism. And this led us to hypothesize that when we layer in FUS, these anesthetics may differentially alter the underlying reactivity of the brain. And this will ultimately produce anesthesia-dependent synergies and conflicts with respect to sterile inflammation, drug metabolism, or neuronal damage. To test this hypothesis, mice were treated with one of the following interventions, either naive, untreated mice, isoflurane, or ket-dex anesthesia, the addition of microbubbles on both anesthetic backgrounds, and the addition of FUS with microbubbles and isoflurane or ket-dex backgrounds. The FUS parameters are given here, and I just wanted to highlight our peak negative pressure of 0.4 megapascals leads to reliable blood-brain barrier disruption by MRI contrast enhancement, but does not lead to any signs of histological damage. We then harvested these brains 6 hours later for bulk RNA sequencing. The results of that study are summarized by this principal components analysis. Each dot on this PCA represents a biological replicate, and they're all plotted along the components that represent the most variance in the data. So very clearly, we can see kind of two distinct clusters along that first principal component. On the right here, all three of these conditions are mice treated with ket-dex, while those treated with various conditions on the left were either treated with ISO or no anesthesia at all, already suggesting to us that ket-dex may be driving a lot of the variation in our data. If you take a look at the pink and purple groups, those treated with focused ultrasound on different anesthetic backgrounds, you can see those tend to trend higher along that second principal component, suggesting that that may be a secondary source of variation in the data set. This is further highlighted by these volcano plots here, where we are doing pairwise comparisons of gene expression. In isotreated mice versus naive mice, we see only a handful of genes that are differentially expressed. However, when we treat mice with ketamine and ketamine dexmedetomidine and compare them to the same naive mice, we see thousands of genes differentially expressed. Again, really highlighting the differences that these anesthetics impart on the brain parenchyma. When we layer on focused ultrasound and microbubbles while controlling for the anesthetic backgrounds, we can see a moderate effect on gene expression on the order of dozens to hundreds of genes differentially expressed. And I'll go into some of the consequences of that differential expression on this slide. So to identify how different sets of genes are correlated with phenotypes in all of these different comparisons, we perform gene set enrichment analysis. And the results of that are summarized on this table here. The rows of this table correspond to different biological pathways. So this first pathway is, for example, endothelial cell differentiation. The columns correspond to different contrasts. And I wanted to highlight the right three columns of both the ISO group and the ket-dex group, as these are the ones associated with focused ultrasound treatment. We were able to identify 6 broad classes of biological function that were differentially affected by FUS and or anesthesia. And very quickly, you can start to see that there are both overlaps and distinct groups that are affected by FUS dependent on the anesthetic background. So for example, endothelial activity, inflammation, and tissue repair were all significantly enriched by focused ultrasound regardless of the anesthetic background. But as we start to look at pathways associated with metabolism, platelet activity, or discrete signaling pathways, now we can see that the anesthetic background actually alters the effect of focused ultrasound in terms of activating or repressing these pathways. So ultimately, we were able to determine that the acute transcriptional responses in the brain are affected significantly by CET-DEX and only moderately by focused ultrasound. We identified common pathways upregulated by FUS that are independent of anesthesia, including those associated with endothelial activation, sterile inflammation, and repair. FUS pathways that were activated dependent on the anesthetic background included metabolic signaling, platelet activation, and discrete molecular signaling pathways. And ultimately, we hope that investigators are able to use this information to design future studies, essentially selecting anesthetic that works best with their intervention or disease model. And we also hope that it will contextualize past studies depending on which anesthetic was used. So I'd like to thank our lab and our collaborators, as well as our funding sources, and all of you for your time. Thank you.